Thanks for staying the course. It's always good when you organise a conference to save a highlight to the end. So it's a pleasure to welcome James DeGregori from the University of Colorado. He just gave me a little lesson on how to pronounce his name, but I realise I haven't done well. I'm sorry, James. But really great to have you talking. And I love your title. It's a bit depressing, though. Somatic evolution, cancer, and our inevitable decline with age, inextricably linked.
Well, thanks a lot. It's a real honor to give this last talk, and I thank the organizers for inviting me. And yeah, no, this, as I was telling people last night, I think the way I deal with aging is to have a morbid sense of humor. So hopefully you'll come along with me on that. And I also should mention that, with the theme of art in this conference, I'll be showing quite a bit of artwork by my son. And he's been doing the artwork for my books and papers and everything for at least 15 years. And so I think he's quite talented. So I'll start with what we already know, which is that about 90% of cancers develop after the age of 50, and that's shown here. And I think the big question is why, and I think this has been, almost thought to be solved in the field of cancer. And in fact, there's been a ton of models that have been used to simply say, well, all you need is the stepwise accumulation of, let's say, 6 events or 6 mutations or something like that. And that just simply by altering the fitness effects of those mutations, you can fit this curve quite well. But as any mathematician will tell you, it's easy to fit a curve. It's another question as to whether it's the right explanation. And so one of the things that we looked at very early is that, we have a lot of diversity in cancers and leukemias that are arising in our body. And, if you look at, for example, everything from CML to AML, they either are known to be driven by about one event at diagnosis, shown for CML here, or with multiple events like for AML and CLL. And the same thing with solid cancers. They vary anywhere from, thought to be driven by one mutation, like for mesothelioma, up to 11 mutations for colon cancer. And that's based on driver mutation rates as determined by, Inigo Martin-Carrena and colleagues in that Cell paper that's already been cited a few years ago. And while these numbers are soft and they don't include large genomic rearrangements and they don't include epigenetic events, I think it's safe to say that cancers are driven by a wide numbers of different events, that not all cancers are driven by about the same number of events. And the other thing to note is that cancers arise in very different stem cell and progenitor cell populations. So we've got about 10 million intestinal crypts in our body. Each one has, 10 to 20 stem cells. So, on the order of 100 million such cells and organized in these crypt structures. And in the hematopoietic system, we know that they're about on the order of 100 to 200,000 hematopoietic stem cells that maintain. And these are thought to be the sources of these different cancers. And yet, despite all of these differences in the driver number and the stem cell pool sizes, we do a very simple transformation. We just take these different cancers and we normalize them to our maximum. This was one of the most striking results that I had seen, and it was from Andre Roshak, who was a postdoc in my lab a number of years ago, is that they fall right on top of each other. And this suggests something that goes beyond simply mutation accumulation that I think we need to consider. And in fact, when Andre did a Monte Carlo model, which I won't go into the details of, but if you like modeling, it's in this paper, he found that in order to generate these curves despite variance in these other parameters, he really needed to include a parameter of differential selection, where the mutation would be variably selected against in youth and selected for in the second part of a life, of a potential lifespan, I should say. And so this raises the question, so why do we get cancer and other diseases mostly at older ages? And in fact, why do we even age at all? And most of you guys, and you just heard Amy introduce life history theory, most of you will already know why this is, and that's because there's minimal selection against disease beyond the age where most individuals would already be dead by other causes. And this is a curve. This is actually just a rough approximation from fossil records from, I think this one is from Europeans tens of thousands of years ago, but I've seen very similar curves for Americans in the, I'm forgetting the mounds of Missouri, but from 1,000 plus years ago, Native American populations, as well as from Eastern Africa, where they've done population studies. So from fossil history, we see that most individuals, not all individuals, are dead by about 50. And I'll note that this underestimates childhood mortality. So if you make it to adulthood, your chances of surviving to older ages is much greater than if you didn't make it to like age 15. Well, actually, that's kind of obvious. But anyway, but what we can see is that this curve has minimal overlap with modern curves of cancer and heart disease. You could graph out death by COVID. You could graph out neurodegeneration. Basically, we mostly get diseases, and we think of them as diseases of old ages, but these are mostly the reasons that we die at ages beyond when most of us would already have been dead by other causes for most of our evolutionary history. And oddly, this is not taught in med schools. Med students do not think about why we get most of our diseases in old age. And so I think it's really quite simple. This clearly says there's a force of natural selection that's acting on this curve. Now, what that force is another question. But this suggests that there's something that's pushing the cancer incidence. And again, I'm saying using cancer incidence almost as an example. This could be heart disease, kidney failure, neurodegeneration. We're pushing this out to the right side of a potential lifespan. And if this were mice, you would just change the scale. And most mice in the wild are dead by 6 months if you track them. So please don't tell me that mice are not good at preventing cancer. They're fantastic at preventing cancer. As you just heard from Amy, it depends on your life history, how you have your babies. If you have a breed early, breed often strategy, preventing cancer at age 2 would have been a waste of energy. So, and for these reasons, even our lives, even under modern industrialized society conditions, is not linear. In fact, from about the age of 5 to about the age of 37, your chance of dying every year is equally low, something on the order of a little bit under 1 in 1,000. So in fact, it's amazing that we can build a machine of trillions of cells and maintain it with such a low error rate for almost, half of a century. I think that's amazing. But starting at about age 37, with every subsequent year, you have an increased chance of dying as in your previous year. As I said, this is going to be a depressing talk. And I like to say we don't just go downhill, we pick up speed, because that's a logarithmic increase. In fact, you can graph it on a log curve known as a Gompertz curve. And so we basically accelerate as we go downhill. And I'll try to convince you today that I think this is really going to require feed-forward loops. And I'll try to present at least what I think is one such feed-forward loop. And this is despite the fact that most measure of damage accumulation, at least from maturity onward, whether we're talking mutations, epigenetic changes, or other changes, is pretty linear. So we're accumulating damage linearly, and yet we're not aging linearly. And I think this is because of different measures of resiliency. And I'll go over these in the very next slide with redundancy, replacement, and repair. So we have redundancy. As I mentioned, for example, we have 200,000 hematopoietic stem cells. If one of them is misperforming, you just get rid of it. You can replace it, right? We have intrinsic maintenance machinery. We have ways of getting rid of cells that are not only that are poorly functional, but that are even hyperfunctional, that might be potentially transformed. They can undergo apoptosis or senescence. And we have immune surveillance. Peer pressure is really interesting, where the cell might even have an oncogenic mutation, but if it's surrounded by normal cells, it actually acts normally. In other words, it doesn't have a transformed phenotype. And finally, one of my favorites is cell competition, where your neighboring cells eliminate the cell that has a mutation that either makes it poorly functional or that makes it hyperfunctional. They might even have a transforming mutation. Really beautiful work that really started out in Drosophila. And this all helps maintain a youthful soma. And this youthful soma then is important for a number of things. Our robustness, our ability to reproduce successfully, tissue function, immune defense, avoiding disease, and also anti-cancer defenses. But I think anti-cancer defense is just one of those mechanisms. As I told you, my son Michael is very creative. I just told him to draw something for anti-cancer defenses, and he shows a broom sweeping away a crab, which I just love. So if we think about this, I think we can think about life as having different sort of phases. And so cancer incidence is shaped by this changing aging-dependent balance of, and I won't get into the drift, but stabilizing selection and positive selection. So what I've been proposing for many years is that during youth, our tissues are essentially acted upon by stabilizing selection. We evolve stem and progenitor cell pools that are functioning somewhere near a fitness maximum, a somatic fitness maximum. In other words, changes in that stem cell will tend to make it less fit, not more fit, even mutations that we would consider oncogenic. And there are many examples of this for the hematopoietic stem cell system. So for example, really nice work from Sean Morrison and company has shown that if you either decrease translation in hematopoietic stem cells or increase translation, you reduce the ability of those stem cells to be maintained in the pool. And many different cancer-associated mutations would cause increases in translation, and many forms of damage to a cell will cause decreases in translation. So it's a beautiful system of maintaining a parameter within a certain window. And this acts through youth, and I think it's facilitated by these tissue maintenance mechanisms that I just went over. But of course, we don't maintain our tissues forever because that requires, for example, energetic cost. Although I don't know if it's ever been proven that energetic cost is the reason, but that's at least the assumption. And so you can imagine with limiting resources, that investment is going to wane at older ages, or it's going to even wane more if we do things like smoking or have a poor diet and so forth. And with that change in our tissue microenvironments, this can lead to positive selection for new types that are now adaptive to this new microenvironment and can promote cancer incidence. And I think that the tissue microenvironment is really key because I think what changes about the stem cell can in part be intrinsic to the stem cell. They can accumulate damage. But the more overwhelming factor is that the tissue microenvironment is decaying, and that changes what is the most adaptive type. So now I'm going to switch to clonal hematopoiesis as sort of an example of somatic evolution happening within our body. And I think we've heard about this already during the talk, but about 15% of us as we get old, and this is largely restricted to the elderly, will get clonal hematopoiesis where a significant percentage, defined as I think over about 4% of our blood cells, will have a clonal driver that is promoting clonal expansion of, it's thought to be at the stem cell stage, not proven, but probably is, that's driving the expansion of hematopoietic cells. And this can be detected in the blood. And it's a precursor to blood cancers. But I'll note, it's not just a precursor to blood cancers, it's a precursor to, it's a risk factor, I should say, for many other diseases, including a number of different cardiovascular diseases, pulmonary diseases. And every month there seems to be a new liver disease or some new disease that's associated with clonal hematopoiesis. And in fact, if you look at, for example, I should go back. This is the odds ratio for AML. So you look at TET2, it has an 8-fold increased risk of developing AML if you've got TET2 clonal hematopoiesis. But this is a cardiovascular disease, is only about 2-fold increased. But if you think about it, given our lifetime risk of cardiovascular disease is 1 to 2 to 1 to 3, and lifetime risk of AML is 1 to 200, the bigger thing you've got to worry about is the cardiovascular disease. That's the bigger factor. And there are a number of papers showing that these expansions can even directly contribute to cardiovascular disease. So I think when we start thinking about somatic evolution, we have to get away from just thinking about the somatic evolution as precursors to cancer. I think they're contributing to a number of different diseases. And so we wanted to ask, well, is there differential selection for TET2? I'd shown you the monoclonal modeling where it looked like we needed differential selection for a mutation in order to avoid cancers until we get to old age. And we've actually shown this before with some other model systems, but I'm going to show you one now for clonal hematopoiesis. And so what we're doing here is we're taking either young or old donors, and the old donors are always close to 20 to 24 months old, about approaching 2 years old. Young will tend to be 2 to 3 months old. We take out their stem cells, their hematopoietic stem cells. It's a highly stem cell enriched fraction. And then Marco, the postdoc in my lab, developed really efficient methods to introduce in CRISPR RNAs. He can basically get them close to 100% of the cells. And he's actually combining 2 pools here, one where he's knocking out TET2 and another one where he does a CRISPR insertion or a CRISPR deletion in a region of the genome that shouldn't matter. but it's still going to make the DNA break, which I think is important. And he combines these, and he combines them at a ratio where there's fewer of the TET2 knocked out ones to begin with, and he puts them into mice. And one important thing is he does a very mild conditioning, only getting a few of the cells in because we basically want to study them in a relatively normal environment under normal conditions. And when he did this, he sees that they expand substantially faster in old recipients when the stem cells come from old than in young recipients. And this just shows you for some peripheral blood populations. And the results are even more dramatic when we look at the stem cell lineages. And these are the true stem cells. And we can see a pretty big difference between the expansion of these cells in young versus old. So there seems to be a fitness advantage that's aging dependent. And we looked across the lifespan, and you can see that it sort of increases with lifespan if you look at peripheral blood from 2 to up to 20 months up here. Middle ages are kind of mixed together. But what's notable is that we really look at the HSCs once we sacrifice these mice, then you can see that we only really get the selection in the HSCs over the long term. And we've gone on to show that by sorting out the hematopoietic stem cells, we only see the selection within the stem cells. And finally, we went on to then show that this fitness is relative. So if we put cells in competition with each other, and I'll walk you through these different competitions, first off, you can see the expected. If we have old stem cells competing with young stem cells, the young ones win. So the old ones are less fit, and they start off even on the first time point. One would be neutral, basically. They basically decline. The old ones are declining in relation to the young ones. We're always looking, what's being graphed is the first in the competition. But in contrast, if we look at old TET2 knockout versus old, we can see that it's got that fitness advantage that I already showed you before. But if we put old TET2 knockout versus young, we get much less of a fitness advantage. So in part, it's more fit because the competition's crappy. Probably not a bad lesson for life if you want to get ahead. But there's still a small fitness advantage even in young competition. But if we look at the old knockout in relation to young TET2 knockout, we can see that the TET2 knockout actually is even more fit if it's in young. So the reason that the TET2 knockout in the old is expanding is not because they're intrinsically better than, for example, a young TET2 knockout. They're just better than the old competition they're having to deal with. And so we kind of put this on a relative fitness scale. And so even though we do see some selection in young, relative, if we put TET2 in, that fitness differential is much greater in the old because the old stem cells at the beginning are so much worse off. So then the question is, why is the TET2 mutation selectively adaptive in the old context? And of course, when you don't have a clue, you do something omics. So we did single cell RNA-seq, and we sorted out young and old hematopoietic stem cells and progenitor cells with or without the TET2 mutants. And this is again Marco doing the work. And we got a lot of help from Lamis and Sheng at USC. They were at JAX before, but they really helped with the bioinformatics. And so this just shows you the different cell types that we're looking at. We actually purposely spiked in some mature cells. I won't get into that today because we want to know how these mutations are affecting the mature cells as well. Because when you get these clonal expansions, it's not just the stem and progenitors you have to worry about, but if they're making abnormal mature cells, that can affect your overall phenotype as an individual. And when we did this, we got what seemed at first to be sort of surprising. In the old, if we just compare older versus young, in both cases, we get this upregulation of these pathways in old that for regulation of ribosomal and OXPHOS genes, stuff that you would think of being as transforming, like MYC and mTOR. So these are up and old. But in some ways, as I told you before, maybe that's not surprising because what has been shown by others is that activating these pathways in stem cells, and they did it in young stem cells, actually leads to the reduced maintenance. So this could be contributing to the fact that they're hyperactive. And it's also interesting because mTOR, of course, inhibition of mTOR is one of the best ways to increase longevity in a number of different model organisms. And so when we look at the mutation effect, so what happens if you knock out TET2, we basically reverse all of those effects. So TET2 is essentially having a rejuvenating effect on the stem cells. Now, if that was all that it did, well, that would be great. We could go on our merry way. But TET2 is also inducing, for example, increased inflammation and other pathways that I won't go into, but it's making the stem cells abnormal. At the same time, it's restoring their more youthful profiles for these different parameters. So what I've shown you this far is that old age promotes selection for TET2 loss in HSCs, essentially helping provide some explanation for why clonal hematopoiesis increases in old age. Somatic fitness is relative, meaning it's not that those cells are super cells, they're just better than the crappy old cells that they're in competition with. These old hematopoietic stem cells exhibit upregulation of ribosomal electron transport genes, and TET2 loss can restore that lower expression. So what are the implications of this? It raises the question is, can we develop interventions that reverse aging-associated increased selection for TET2 loss? A lot of the interventions that are being thought of for clonal hematopoiesis are targeting the actual clone, the mutant clone. What I'm suggesting is that if we could reduce selection for the clone, that might actually be a more productive strategy. Another question, would such an intervention not only reduce the risk of leukemia, but also other consequences of TET2 mutant blood cells, such as for cardiovascular disease? So it could have other benefits as well. And we're currently testing some different interventions. So I actually want to take a quick break here. I know I'm only halfway done, but are there any, because honestly, no one really has a 30-minute attention span. So any questions from students or postdocs?
Thanks. Very interesting. Probably you're aware of this study that proposes an inverse relationship between heart disease and clonal hematopoiesis. So not really suggesting that clonal hematopoiesis is driving heart disease, but vice versa. So
inverse relationship in what way? I'm sorry, Ms.
Between heart disease and clonal hematopoiesis. So claiming that it's not clonal hematopoiesis that increases the risk for heart disease, but vice versa, that the heart disease kind of fosters clonal hematopoiesis.
In fact, all we can say right now is there's an association. In fact, actually, you could imagine that, let's say clonal hematopoiesis is promoted by basically a degraded soma. In that case, it would be a marker of a degraded soma, and the degraded soma could be increasing other disease risk. The only thing is they do have mouse data showing that when they induce clonal hematopoiesis in a mouse, that it increased progression of a model of cardiovascular disease. So they do have functional data, and they show that the macrophages that went to the heart or wherever they were going, I don't remember the details, were actually pro-inflammatory, and they had many of the effects that would be expected to promote clonal hematopoiesis. And I think they could even block it by blocking interleukin-1. So that there is some functional data, but I would agree that overall, all we've got is an association.
Thank you.
Yeah. Anything else? Non-students, postdocs? There was one up there, yeah.
Thank you for the talk. I was wondering about the graph you show with cancer incidence going up with age. On the bottom, there's positive selection pushing up at the cell level, I imagine. From the top, there is stabilization selection pushing down at the individual level, I imagine. So at the host, at the human level. And you mentioned the reason why this selection gives up and the cancer risk starts to go up at old age would be an energetic reason. So it's a form of antagonistic pleiotropy. So we save energy by not maintaining these mechanisms because we'll be dead by then.
Yeah, well, it could be that this is an investment that's happening throughout our life, right? In terms of the body you build and maintain. It's the maintenance. In other words, would the energy be better spent on reproduction versus on maintenance?
Yes,
and we've seen this dichotomy a number of times in evolutionary biology. And actually, I would say the stabilizing selection is at the level of cells and tissue. In other words, a cell that in youth, we favor the cell with the evolved type, the evolved phenotype. Change in phenotype tends to be disadvantageous at the cellular level in youth, is what I'm proposing. And at an older age is because of the sort of degradation of the tissue, which we of course knows happens, those of us beyond 37 are well aware of this, that decline in our soma is now promoting positive selection for new phenotypes that are adaptive to the new environment.
But is there evidence for that? Because I've always thought positive selection at the cell level is always equally intense, and there is just an evolutionary pressure on our species to fight this thing until we're about 35.
So,
But
positive selection is always, in what context would that be? I mean, of course, in the real world, we know that positive selection is incredibly context dependent. That's why we've had evolution over millions of years.
Yes,
right?
but I mean, at the single cell level, positive selection is just increased proliferation, as in what the cells want to. I mean, selection will just be increased probability of living offspring for a cell that is just increased proliferation. That's always going to be advantageous to a cell because the number of copies of the genome will increase.
Yeah, if a cell was in a tissue culture dish, I'd agree with you. But stem cells actually have a very special case, and it's, I think, a beautiful thing about stem cells is that too much proliferation of a stem cell is the best way to drive that stem cell to differentiate. So, in fact, this is seen, in our model system. We actually show that it's not equal, right? That in the young, we do have much less positive selection than the old.
My point is that the stem cell is somehow programmed for that to happen. So there is some kind of regulation so that the stem cell gets out of control in youth, it will differentiate.
Yeah.
But that is caused by a mechanism which is kept in place by selection at our species level.
Oh, of course.
That's what I mean.
No, absolutely. Yeah, no, of course, all of this is kept in place because of evolution at the species level. These are all mechanisms that we evolved
So
not die prematurely of cancer.
My point was actually something else. So it's antagonistic pleiotropy. So we save resources in youth by neglecting, to some extent, maintenance in favor of reproduction. There's a theory. But I've always thought it's not really necessary. So imagine that is not actually the case. Imagine there's just no energy saving, or even we are losing energy by doing this. It will still happen because there's no leverage for selection to do anything because there's not enough people alive. So just by drift, you won't be able to maintain this thing, even if there is no energy saving, even if we are losing energy by doing this. Selection can't do it. There's just no individuals alive at that age.
Yeah, that's part of the theory is that the reason, I mean, this is not our theory, this has been going back for Menawar and others for 70 years, but is that there's a waning of the selective pressure to maintain us because there are few individuals even around. So these ideas of programmed aging, I think, fall apart when you try to think of why would mice age, and they're aging way after you're never going to find a mouse of that age.
Yes.
So in some ways, we actually have maintenance systems that take us beyond what we would, we're lucky that we stay as healthy as we are into our 60s and 70s.
But remember, I've always seen that
and I should probably move on.
Yes, I know. The disposable soma.
Let me move on, and then we'll catch the others at the end. Otherwise, I don't want to head over, okay?
Yeah. Thank you.
Sure. So I'm going to now get into somatic evolution within solid tissues, which we've heard a ton about at this conference. And, and I think probably half of these papers or half of the papers that are out there on this came from this campus. But, we know that cells with cancer-associated mutations completely overtake our tissues as we age. And a postdoc in my lab, Ed Evans, did sort of meta-analysis across all these different studies, at least that were studies back then in 2021 or 2020 when he was doing most of these. And what he could see is that across these tissues, in many cases, as we get older, we're approaching close to 100% of the cells of the tissue. And you've seen this from this famous now schematas that I think has been shown about 500 times at this meeting, showing that these clones can completely overtake the esophagus. But this varies quite a bit from tissue to tissue. As you can see, the y-axis is quite different for different tissues. And so if he adds them all up, and this is again just the tissues that have been sequenced, and just at the limited depth that they were being sequenced at, which in most cases this was based on like whole genome or exome sequencing, so a couple percent, only if it was a couple percent of that piece of tissue could you see it. But most older cancer-free individuals possess at least 100 billion cells that harbor at least one oncogenic mutation by the time you're 60. And yet, only about 40% of us will develop cancer, and that will almost always arise from a single oncogenically initiated cell, although some recent results that are kind of questioning that. So maybe at least initiation might be polyclonal. So then the question is, what do the other 100 billion or so cells with these oncogenic mutations do? And I'm putting oncogenic mutations in air quotes very much on purpose. I think we need to stop thinking about these mutations as really oncogenic. Well, they can be, but they're certainly not always. And so to really study this, we have this wonderful collaboration with Charlie Swanton, Miriam Jamal Hanjani, and many other people. A lot of the analysis has been done by Amelia, and a lot of the recent analysis has been done by Faz, who you just heard about yesterday and presented some of this. And so we're going with 4 different cohorts here. They're either from bronchoscopies. These are people that had a detected lesion, but in half the case, the lesion turns out to be a false positive. And we are always getting the piece of tissue from the other lung, from another lobe, so it should be completely disease-free. We've got tracer X samples, and these are near a cancer, but I think at least a few centimeters away from the cancer. And then the piece samples, these are people that died of cancer, but we are mostly looking at non-lung cancers, and at least the piece of tissue we take has been seen to be disease-free. And we also do some piece multi-region samples that I'll get into. And we've heard about duplex sequencing. Yes, I know it's the ugly cousin to nanoseq, but it's still, for our purposes, I think it's plenty sensitive. And the bottom line is that you independently capture and sequence the 2 strands of DNA, and thus you only count a variant if it's seen on both strands, which greatly reduces the error rate. And so we're sequencing to 10 to 20,000 duplex depth, which means sequencing to almost a million-fold depth if you just talk about Illumina sequencing. So this is what our patterns would generally look like. So you can see allele frequency here on a log scale going up. And so higher variant allele frequencies are up here. And you can see, there's definitely more mutations in the smokers than the never smokers. But overall, you see plenty of clones whether people smoked or didn't smoke. And by the way, we're covering about 30 genes in our panel, and these are the genes that are being shown here. And so this is showing you everything, which if I tried to go into, I'd burn all the rest of my time. But these are the different cohorts that I talked about, and these doesn't include the multi-region sampling. But you can see that we get up to over 1,000 mutations, but mostly in the few hundred mutation level. The red ones are cosmic designated. So these are in the cosmic database as being found in cancers. And if you just look at the cosmic mutations, you can see that a lot of the top ones are in P53, PIK3CA, KRAS, and so forth. And you get some down here that have pretty low mutation counts. And I'll note that TIAM2 here we consider that a control gene, and it has very few mutants. So most of the mutants we see are actually in the cancer genes. And I recognize it's a limitation of this study that we selected cancer genes for the study. But that's because we could never afford to do whole genome or even whole exome yet. And so what we can see just right off the bat, and this was based on the bronchoscopies, and of course, people coming for bronchoscopies are for CT scans are mostly smokers and former smokers. And so we get very few never smokers, but the few that we got, we noted that we've rarely ever got high variant allele frequency for variants. While in the smokers, you get lots of higher VAF variants. So there's more selection for these variants in the people that have smoked than in the never smokers. And they're in a lot of the genes that I've already talked about. And we see the same thing with indels as well. The indels are pretty predominantly seen for Notch1, but we see it in some of these other tumor suppressors. It's notable, and it's kind of a nice check on the data, that we don't see indels in things like KRAS or PIK3CA, or we see very few of them, really suggesting that these indels are being selected because they're disrupting these genes. And so I'm going to focus a little bit on KRAS for a little bit. And so one thing we notice is that there's significantly, not only are there more variants per individual in KRAS if they were ever smokers than if they were never smokers, but the variant allele frequency is significantly higher. So there's not just that smoking might be causing mutations, but it's selecting for the mutation. And I should mention the very fact that we see it is probably because it was selected for. So it's really hard to know how much of a contribution is the causation of mutation, but I think the more clear thing is we're clearly selecting for these variants. And we see the same thing with EGFR. And if we look at these variants, what we see is this kaleidoscope of variants across these, and these are all cosmic designated. These have all been seen in a cancer. These aren't just any variant. And yet, if we look at non-small cell lung cancer, there's really 4 variants, maybe 5 if you count G13C, that really dominate what happens in actual cancer. And yet we get this large array here for these variants. And so these are just the 5 variants shown there. And I'm graphing this a different way now, where now if we look, we can see that even in the smokers, which are these first 2, we see that most of the variants just shown here in gray, and all of these again are cosmic designated variants. Most of the variants are not the ones found in the actual lung cancers. So they're causing clonal expansions, but they're not leading to cancer. So just activating RAS doesn't appear to be enough. It might be how you activate RAS that matters. And you can contrast this for the actual tumors where they're, again, dominated by the top 5 mutations, right? And we'll also note that the never smokers, particularly in these 2 cohorts, we pretty much never saw in the BDRE or the PEACE cohort, we saw 0 of these actual cancer, lung cancer associated hotspots, the bad boys. We did see a couple in TRACERx and I think 2 individuals, and there might be reasons for that. You have to keep in mind these are close to the cancer and so forth. But so we seem to do a very good job, if you don't smoke, of preventing the really dangerous mutations. And this is just graphed out in another cases, it's a % of cases. So cancer is along the x-axis and normal is along the y-axis. And if you look in the smokers, you indeed do select for the sort of the dominant high-risk alleles of KRAS that are seen in cancers. But you also get a lot of these variants that are rarely seen in cancers. Every once in a while they're seen in cancers. But for the never smokers, we rarely ever see the really dangerous ones, but we do get some of the not so dangerous ones that are rarely seen in cancer. So if you want to avoid selecting for lung cancer associated KRAS mutations, don't smoke. And we see this across other genes as well. And there's a non-equivalency between what's promoting clonal expansion and what is actually leading to cancer. And so everything starred here is significantly greater in the normal tissue, shown in blue, relative to what's seen in cancer. And we're limiting all variants here to actual cosmic mutations. So you can see, for example, we get a lot more PIK3CA expansions than we do in the cancers. But what's really interesting is the stuff down here. These are genes where their mutations will cause cancer somewhere in your body, but perhaps not in the lung. And so you're getting different selection against these because they're not maybe as dangerous for the lung, at least in terms of cancer. And if we look at a particular gene, it even gets more interesting because now what we see is that, for example, with PIK3CA, about 80% of cancers have one of 3 variants here, shown the high peaks basically. But if we look at the variants that we're seeing in the normal tissue, it's much more spread across the protein. And anyone that's in red has actually been seen in the cosmic database. So it's been associated with some cancer somewhere, even if rarely. And we want to know, well, what are these other variants even doing? Are they, could they actually be having some effect that's similar to these hotspot mutations? And indeed, we worked with my colleague Alain Eisenmesser, and he modeled these onto the structure of PIK3CA. So the PIK3CA protein encoded here is the kinase subunit, and the green is the P85 regulatory subunit. And what you can see is the red mutations are those true hotspots, but the yellow ones, which are the other mutations that are found at least a couple of times in the normal tissue, are mapping to this similar interface. So what's purely speculative at this point, we think these might be also disrupting this interaction with the regulatory subunit. Perhaps not in the same way that those hotspots do, but enough to promote clonal expansion within the normal tissue. And I'll just note that for EGFR, we get a similar phenomena where the real hotspots in lung cancer are less represented in the normal tissue, but you're still getting other variants that are seen in some cancers somewhere, even if rarely. So just some interim thoughts that only a small subset of mutation-driven clonal expansions have a likelihood of contributing to a cancer in the tissue that they're found. And a mutation can be adaptive in a normal tissue, but not contribute to malignancy. I mean, in some ways, we already know that from the work with Notch1, for example, from Phil Jones's lab. And clonal evolution in a normal tissue does not equal cancer evolution. And so I don't really need to introduce DNDS. This is a great thing about giving the last talk. All these concepts have been introduced already, but non-synonymous changes relative to synonymous changes in coding sequence. And so working with Faz and Amelia and his mentors, Amelia and Alex, basically Faz came up with a strategy to somehow look at DNDS while also contributing, understanding the contribution of variant allele frequency. Because the higher variant allele frequency should also indicate increased selection. So he basically calculates DNDS and calculates essentially a number of excess drivers that are seen. And you can see that they're in many of the genes that we're also seeing lots of mutations with. And then he calculates the VAF selection coefficient by taking into account the VAF driver, the VAF driver selection. And in combining these, he gets a VAF selection coefficient. And sorry, that's as much as I'm going to be able to explain it at this point. But essentially, it's taking into account both excess driver variants and their variant allele frequency. And what you can see is that we're getting a lot of selection on NF1, P53, ERBB4, and Notch1. And we see this selectively in the smokers, which much less in the never smokers, which is shown in blue. And this is for the Tracer X cohort, but we see the same thing for PEACE and BDRE, where we see P53, Notch1, NF1, and ERBB4 are really the top selected genes in terms of their variants. And I'll mention this doesn't take into account indels or stop codons. So we can also consider those when we just look at variant allele frequency. And what you can see is for these tumor suppressor genes, if we look at indels, there's significantly greater variant allele frequency for the indels in the smokers than the never smokers in each case. And for P53, we also see this association with point mutations, but of course we know P53 is frequently disrupted by point mutations. I'll note that if they're red, they're cosmic designated, but for these tumor suppressors, we often don't see a lot of cosmic mutated indels because there's so many ways to make an indel that you're unlikely to see it over and over again, but we do know it's clearly disrupting the protein. So now the question is, well, just how prevalent are these mutant clones within our lungs? And so if we do what we call a sum of VAFs, where we take all the VAFs in all the mutations in that gene in one particular individual, and we sum them up. So that's sort of how much area of that little piece of tissue was occupied by that particular mutation. And when we do that, we could see that for many of these different tumor suppressors, that calculation, and this includes everything that's protein disrupting, including indels and stop codons and SNPs and so forth. If it disrupts the protein, we included it. And you get more in the smokers than the never smokers. And for each of these tumor suppressors, as well as for some genes that are activated by more sort of point mutations. And I'll note that, one thing I should note that looking at the allele frequencies, they tend to be on the order of 1 in 1,000 for most of these genes. So these would not have been picked up by a lot of traditional sequencing methods. And for the control gene, TM2, we see that the mutations are more on the order of about 10 to the minus fourth. And we'll note that we even covered more of this. We had a lot of coverage of TM2 in our panel. A couple of kilobases of the 50 kilobases was covered by this. So this sort of suggests that it's kind of weak evidence that most of the mutations that we observe in the CGC, cancer gene consensus genes, are likely under positive selection, that there's a reason we're seeing those variants. And if we combine all genes together that are in our panel, remember this is just limited to what we looked at, so this is probably, again, a gross underestimate. We get on the order of about, about 2% for the never smokers and about 9% for the smokers. And if we think that most of these variants are heterozygous, then that would mean that somewhere between 4 and 18% of your lung, depending on whether you smoke, is occupied by these clones. This number, I think, would be much greater if we included, the whole genome in our panel. If someone wants to give me the money. So now, in the final couple minutes, I'm going to turn to the multi-region studies. So this is where, in the PEACE cohort, we got up to 30 different small pieces from the same individual. And this is really thanks to the collaboration with Mariam Jamal Hanjani and her colleagues. And so this just shows you the 7 individuals that we'll be looking at. But what's interesting, if you just look at their total mutational pattern, you could see clear differences between these different individuals. Like, for example, this person just has tons of positive selection across the different regions, or tons of high VAF alleles, I'm assuming, are into positive selection. But we also see that the dominant gene that's being mutated the most tends to vary in the different individuals. And we can also see this if we look at it this way, and this is just showing the dominant gene that's being, or the dominant variant for a particular gene in each individual. And so, I'm sorry, here are the genes going up and down, and these are just 3 of the individuals showing you as an example. And you can see that there's differences between the individuals in terms of which genes are being selected for. And just to focus on PIK3CA, and each color is a different variant if your eyes aren't good enough to see the different variant that's actually written within the circle. And the size of the circle represents its variant allele frequency. And you can see this individual, really almost every region ended up with an activating PIK3CA mutation. So there seemed to be strong selection across this individual. And I should correct myself, I don't know if they're all activating. They're all found in cosmic, meaning they're found in some cancer somewhere. That doesn't prove they're activating, but I bet they are. But you can see there's variance across these individuals and what's being selected for. And what Amelia did that was really kind of cool is she basically created PCA plots based on variant allele frequency and mutation identity. And when she does that, you see that for any individual, all of their regions cluster. So there seems to be a certain flavor of selection throughout the lung for any particular individual. And this person, even over here, we had both left and right lungs for this person, and they even cluster together. So it even goes beyond a single lung. So I find that really fascinating that there's, person-specific. I actually doubt that's due to their genetics. I bet that's due to exposures. And one caveat to all this is from the PEACE study. Many of these people had cancers, so they've been treated. So this may relate more to their treatment than to sort of even their life history or their history of their life. I shouldn't use the word life history. So in the final couple minutes, we'll ask, what are the consequences of these mutation-driven clonal expansions towards lung function? We've been thinking about them as cancer-associated mutations, but I think it's important to consider, as I mentioned, we've got 100 billion of these in our body by the time we're older. What are they doing? I mean, these are genes that are important not just for being mutated and causing cancers, but these are genes that are important developmentally. They regulate differentiation. They regulate many different cell fate decisions. What happens when you disrupt them? So to do this, we have been using a mouse model. So we can, through intranasal instillation, we can introduce guide RNAs that disrupt NF1, our control region. And then we can smoke the mice for 6 weeks. They're in a smoking chamber, so they're exposed to cigarette smoke. And then we let them recover for a week, and then in this case, we harvested the mice and digested the lungs. And what we found is, indeed, with smoking, we see selection for NF1 crispring, which is one of the most frequent mutations we find in the normal lung of individuals that was selected for. It was increased in frequency and variant allele frequency with smoking. And the same thing for RAS mutations, which we also saw specifically in the smokers. And so they're increasing in frequency with smoking, but even more importantly, I think, is that if you look at, these are the control cells, and you can see that they have different differentiation statuses. But if we knock out CRISPR, we seem to really be majorly sticking with this one population. Other studies seem to show that this is more of a progenitor population, but we haven't proven that for these particular cells. So take that with a grain of salt. And just that control non-targeting shows a very similar distribution. We had fewer of the cells because they weren't selected for, but you could see it's definitely not this pattern. And this just quantifies it. You can see this selection in each case for NF1 and RAS with smoking. But what's the functional consequence? So when we look at these control cells, we're looking at a measure of permeability of an air-liquid phase culture. So what I just showed you was mice. Now we're in culture, air-liquid interface cultures. Not as cool as epithelioids, but still useful. They form a very nice multi-layered epithelium. You can see that if you expose them to cigarette smoke extract, you get a decrease in permeability. More of this FITC is passing through. And if you give them NF1 CRISPR, it's not significant in terms of the change, although it looks like it's trending up in terms of disrupting the monolayer. But if you combine those two together, we get a synergistic effect. So having these expansions and smoking may be significantly worse than just having the expansions. So now I can sum up. So I've shown you that smoking promotes selection for oncogenic variants in the lung, or cancer-associated mutations, I should say. And this clonal selection is evident even in the lungs of never smokers. We observe pervasive positive selection on a wide range of mutations in every tissue biopsy. So our tissues are basically active laboratories of natural selection. What I found really fascinating is that this adaptive landscape is specific for an individual's lung, demonstrating repeated convergent evolution. And finally, our preliminary studies indicate that these clones are negatively impact lung function. And so this leads to this positive feedback model. I mentioned, why is it that we not only go downhill but pick up speed? So you can imagine that with aging and smoking, you're perturbing tissue. And this tissue perturbation can lead to these harmful clonal expansions. But these harmful clonal expansions, such as by disrupting barrier function, which should increase inflammation and so forth, are leading to more tissue perturbation. So we're just getting the cycle of decay. But the hope is that we might be able to have interventions here that can end up reducing the risk of this decay towards lung disease. And if we think about this again on this curve of life, that when we're young, I think there's a positive feedback loop that actually maintains our tissue. Meaning healthy tissue impairs these clonal expansions. And if you don't get the clonal expansions, you don't get further selection for the mutations. But that gives way to this positive reinforcing loop later in life. And we can influence this with our lifestyle, our exposures, and maybe our underlying genetics. And what I think is an important point is this is not just a problem for cancers. I think it's a problem for many other diseases. And it may be more universal in terms of its effect on aging. And so just to sum up, I kind of thanked people along the way who were involved in this work. We've got great collaborators who help us get tissues and so forth on our campus, bioinformatics collaborators as well. We have our fantastic collaborators at the Francis Crick Institute and UCL. The patients and their family for donating these tissues, and I mentioned our collaborations on bioinformatics with the single cell work. And of course, Michael did all the artwork. He did the artwork for my book, and he's been doing my artwork for many years. And if you want to hire him, here's his Gmail. And I just want to give a 2-second plug for the Evolution, Medicine, and Public Health. Fantastic journal. I think a lot of the work being done here would be very appropriate for this journal, so consider it. And also, I'm one of the editors-in-chief of Aging and Cancer. And if you have stuff that's related to aging and cancer, please contact me. I probably can even get you a waiver for the cost. And with that, I'm happy to take any questions. Thank you.
Terrific, thank you. Time for a few questions.
Hi, I wanted to ask, and sorry if this is an ignorant question, but I believe early on you said that the curves were best fit if you had negative selection against these mutations in young and positive in old. But with the TET2, it seemed like you saw just weaker positive selection in the young. So do you think that's different across different mutations contributing to that?
I should actually say we actually could model it just fine by having neutral selection and then positive selection later. So the differential was the most important thing. It was the best fit if we had negative selection. The one thing is that you could imagine natural selection could take different strategies, and how much it pushes down on something might depend on just how dangerous it is. If the strategy of letting it be weakly selected, if that still kept it to largely an old age, which it clearly does, it would be a successful strategy. So it's either that or our model system isn't perfect, because we are still giving some exposure to mice to get the cells in there. So that's the other possibility, is simply the model system's not perfect. I think the more important thing is that it's differential. Yeah.
Beautiful talk, James.
Trevor?
Beautiful talk, James.
Oh, there you are. Okay.
Could you comment if there's a role for the immunogenicity of the mutations and how that changes through life?
It's a great question. I mean, I've thought about this a lot, and I think the little that's been done, I think Inigo told me that, I don't know if he's published it, but that they They did not see loss of immunogenicity when they look at mutations within the normal tissue. And don't quote me on that, or don't quote DeGregori on that, because this is based on my memory, which is aging. But I'm willing to bet that there's not much calling. I think there's so many of these variants in our tissues that I think that the immune systems takes a laissez-faire approach to them and just ignores them, because I think that's safer than the alternative, which might lead to massive autoimmunity. I think that's when they start progressing down the road towards malignancy and they start disrupting tissue, that's when the immune system takes notice. That's purely speculative, but we have to go really and look at some of the algorithms and ask that question, I think, more carefully. Unfortunately, I have to check if we even have the HLAs for all these individuals. That would be something that would be important.
Could I also ask really quickly why you had a picture of a dog sunbathing on your DNDS slide?
I was showing off my son's art. Those are my dogs, and my son has done this thing where he every Christmas he gives me pictures of my dogs, but he's humanized them. And it's funny because they're all three females, but he always puts the largest female as the male. So, yeah, Elizabeth's back there. Oh, there you are. Okay.
Hi.
Yeah.
Thanks for the wonderful talk. I noticed that quite a few of your genes are tumor suppressor genes, so you'd expect both copies most likely to be lost or mutated in order to get an effect. Do you have any speculation about that, or is there any way you can disentangle that?
Yeah, no. With our data, we can't. I mean, of course, with the esophageal data from Phil and Inigo, they did do additional sequencing to show that quite a few of the notch mutations were biallelic. But we have no way of knowing that. I mean, we're just getting a gamish of DNA, so we can't tell whether any of these are biallelic. But I think in some of these cases, in some experimental systems, they have shown that some of these variants could have dominant effects, at least haploinsufficient effects. But yeah, I can only speculate.
Can I just ask you about the rate of aging between and within tissues?
Yeah.
And I was thinking about the competition, which is really interesting but hard to measure, right?
I think that, there is that question of whether there's some tissues that are sort of our weak points, right? That their aging is more important than others. I think the hematopoietic system is one of those because it has such general effects. But I don't know if anyone's done a sort of comprehensive analysis of that in terms of, and this wouldn't be relevant not just to even self-renewing tissues, but to non-self-renewing tissues like the heart.
Yeah.
Whether there's a weak point or whether natural selection has sort of balanced that by not making any one tissue the weak point, that we're sort of decaying in unison. And that's, again, pure speculation.
Great talk.
Okay.
So my question is actually about how the work that you've done sort of as a whole relates to this idea sort of, in popular science, in, people's sort of, lay people's thoughts about aging, that you're not necessarily, like, chronological age and biological age might not be the same thing.
Well, sure.
So, I mean, it seems like there's obviously a lot of connections here, but I was wondering whether you could maybe explicitly kind of lay out where you think that idea of biological and chronological age not mapping onto each other fits with this versus, are there limits to sort of how much you can disentangle biological and chronological age? Yeah,
I mean, when you look at aging, death curves, for example, mortality curves, at any given point, that's the population average, right? So there are going to be people that live to 100 and there are going to be people that die at 55, right? So, we have to take that into account. And obviously, our lifestyles have a big influence. And we know you're going to get a leftward shift from smoking. That's an easy one. We know exercise increases longevity, good diet, all the things that grandma told us, right? We know will change these calculuses. Now, they're all averages. You could lead the perfect life and still die of lung cancer at 30, but you're much less likely to die of lung cancer at 30. So I think that there's definitely, and people have developed all kinds of means to track, epigenetic clocks, for example, or biological age, and those seem to be quite accurate. And they've shown that there's quite a big influence of our life history, our experiences through life and our lifestyles in terms of affecting those clocks. I'm not sure if that's answering your question, though.
Well, maybe could you explicitly sort of connect this to like your idea of the compromised tissue? Like, can you measure that with epigenetic? You know what I mean? Like, can we bring these sort of different sort of perspectives, angles together to maybe get more leverage on these questions?
Yeah, no, I think that, I mean, apparently they can measure it epigenetically because when they've looked at, epigenetics of tissue, they do very predictive of how long you're going to live and whether you're going to get disease. So they can be quite predictive. I think things like clonal hematopoiesis, as we discussed earlier, is one of those predictive factors. In fact, it's been shown to be a very strong predictive factor, particularly if you include it with the epigenetic clocks. That's work of Sid Jaiswal and others. So I think it's still not clear whether it's the epigenetic change or the clonal hematopoiesis that is causing the aging, or whether they're markers of an aging soma.
Mm-hmm. Well, and your work would suggest that maybe one of them is measuring the compromised tissue while the other is measuring the consequences of that.
Right.
You could have a feedback loop.
Yeah, no, I think you definitely, I mean, in some ways, if we had good measures of a compromised tissue, we could look at that. And, I think they could probably do that with lung function, for example. There are functional tests of different tissues that we could look at, and I would be shocked if they didn't correlate with our decline. I know that with lung function, it clearly does. It starts going down even in our 30s.
Hi.
Oh, where was
No,
it?
go ahead, carry on. Yep. You've got the microphone.
Oh, there you are. Okay.
Yeah. So I just wanted to ask, did you only consider smoking as the parameter, or did you also look at other points in the metadata, for example, comorbidities? And there could be a difference in the variant allele frequency with the sex, related to the sex, like males and females separately.
Yeah, we haven't seen dramatic effects of sex. And we're looking at that more closely now that we've got the full data sets. When we looked sort of an interim, we didn't see it, but we need to look again. We are looking at other factors, particularly for the PEACE cohort, because they were treated. And so we're now starting to actually dice up the data. Of course, always the problem is that when you start dicing up the data, you start losing power. But we're definitely looking at other factors, particularly factors like, gender, that would be more, have a sex that would have where we have clearly big cohorts because we're pretty equally divided between them. And age is something we've looked at. Really, we only see an age association with variants in the non-smokers, which is kind of interesting. But of course, most of our individuals are between the ages of 55 and 80, so we don't have a full age range.
What about anti-inflammatory drugs? So
I don't know if we know if they've taken anti-inflammatory drugs. That was the other question.
So even in the non-smokers, were there no comorbidities associated with the patients?
We're looking through the data. I think it depends on the cohort, but I don't know how much we have in terms of comorbidities. I have to look and see what we've got in there. But we are going to look at that, but we're limited by the data that we have.
Thank you.
Okay, last question.
Thank you for the talk. I thought it was really intriguing what you said about smoking not only influencing the mutation rate, but also the selection pressures.
Yeah.
And I was wondering whether you think ultimately it's the same between aging and smoking, that it's not so much the mutation rate maybe that changes, but that just the other cells, the healthy normal cells, aren't doing as well.
Yeah, no, clearly. I mean, that's what I showed for the hematopoietic system with the TET2 mutations. I mean, that's part of the model, is that in that sort of, as our tissues decline, our progenitor cells are less well adapted to their tissue environment, and thus you select for change. And I would argue that only a part of that change could promote cancer. That a lot of this selection is independent of that. So I think we've seen from some of the other talks that have even happened in this conference that these clonal expansions in our tissue definitely go up as we get older. But I think that's not just because of time, that's because of changes in microenvironment mediated selection.
Great. I think we'll have to leave it there, but thank you again for a terrific talk.